students, welcome to the Swayam course Microbial Technology. In the previous lectures, you have learned about the genetic system of retroviruses and various kind of viruses. And today we'll be studying about genetic system of yeast, where we'll be exploring the fundamentals and the applications of genetic system of yeast. I am Dr. Richa Sahu, Assistant Professor at University Teaching Department, Chhattisgarh Swami Vivekanand Technical University. So before going to the genetic system of the microorganism that we are talking about, that is yeast, let's first understand what is yeast. So yeast is a type of unicellular fungus that reproduces by either budding or binary fission. So before going to detail, let's talk about the division or reproduction of yeast. So yeast division primarily occurs through a process called as budding, which is a form of asexual reproduction. Here is the breakdown of how this budding works. It starts with the initiation, where the yeast cell begins the process of budding when it reaches a certain size and the internal conditions are favorable. Then the bud formation, a small bulge or bud start to form on the surface of the parent yeast cell. This bud is essentially a smaller new yeast cell that will grow over time. Then the growth of that bud, the bud continues to grow in size while the parent cell also continues to grow. The nucleus of the parent cell will eventually divide and one of the daughter nuclei will move into the growing bud, followed by the maturation of that bud. As the bud grows, it will start to form its own organelle and cellular structures, eventually becoming a full-sized yeast cell. Then separation. Once the bud has matured and is sufficiently large, it will separate from the parent cell, becoming an independent yeast cell. This method of asexual reproduction allows yeast to rapidly proliferate under favorable conditions. In some cases, yeast can also reproduce sexually, where two yeast cells fuse and undergo meiosis to produce spores, which can help the yeast to adapt the changing environmental conditions surrounding it. We will be talking more about in detail about the reproduction of yeast in the forthcoming slide. Now the types of yeast. There are different types of yeast. Let's talk about the first one, the Saccharomyces cerevisiae, commonly used in baking, brewing and scientific research. Another one is Saccharomyces pombe, used in research, especially in studies of the cell cycles. So why we are talking about yeast, or why we are studying about yeast and its genetic system? Because it is very, very important in research and various kind of industry. It is used as a model organism in genetics and cell biology. The industrial application include fermentation of our alcohol production and in the baking industry. So why we used yeast as a model organism? Why yeast? Because the first reason, it has short generation time. Yeast cells divide rapidly, making them an ideal model organism for genetic studies. It has a simple eukaryotic structure. Although simple, yeast cells share many essential biological mechanisms with higher eukaryotic cells, including human. That is why it is an ideal model organism for the studies that are related to humans. Genetic trackability. Yeast is easy to manipulate genetically with well-established method for gene deletion, overexpression and mutation, which we will be studying in the forthcoming slides. Relevance to the human biology. Many genes and pathways in yeast are conserved in humans, making it a powerful tool for studying the basic biological processes and diseases. Talking about the cellular processes, yeast is used to investigate a wide range of cellular processes such as cell cycle regulation, DNA replication, repair, transcription, translation, and cellular signaling. Many fundamental cellular mechanisms were first discovered in yeast only. Why it is very relevant to human biology and its research? Many genes and pathways in yeast are conserved in humans, making it a powerful tool to study basic biological processes, diseases, and to understand pathway behind it, the mechanism behind it, and finally to discover a therapy or drug relating to that disease. Protein expression and function of yeast. Yeast is often used for the production of recombinant proteins. The yeast expression system can produce proteins with post-translation modifications similar to those in higher eukaryotes, making it useful for studying protein function and interaction. What we are talking about right now is why yeast is used as a model organism. Why is it an ideal system to study for research or for an industrial application? Talking about drug discovery and development, why yeast is used? Yeast can be used in high throughput screening assays to identify potential drug candidates. Its use in the drug discovery includes studying drug targets, identifying drug interaction, and understanding the drug metabolism. In biotechnology, yeast is employed in various biotechnological applications, including fermentation process for the production of alcohol. It is widely used in a brewery industries because it can be used for the for the production of alcohol, for the production of breads, for the production of biofuels. Research in yeast helps in improving these industrial processes and develop new application, improve the yield, and obviously reduce the cost. For the drug research and the disease research, yeast is used to model human diseases including cancer, neurodegenerative diseases, and genetic disorders. By introducing human diseases genes into yeast or studying yeast homologs of human disease related genes, researchers can gain insight into disease mechanism and potential therapeutic targets. It is also used for evolutionary studies. How? The study of yeast can provide insight into evolutionary processes and the conservation of biological pathways across different species. This can help in understanding how evolutionary changes affect cellular functions and contribute to disease. Now talking about the structure, before going into the genetics and details of the signaling and pathway of the yeast, let's talk about the structure. The yeast structure is very simple. It has a cell wall, as you can see in this image. It has a plasma membrane followed by a big vacuole, a mitochondria, nucleus, and Golgi apparatus. Now what the cell wall does? It is a basic eukaryotic cell, so it is composed of glucans and manoproteins providing structural integrity and protection. The nucleus of the yeast contains the yeast genetic material or DNA organized into chromosomes. The mitochondria, as we say, the powerhouse of the cell, it is involved in respiration and energy production for the yeast cell. The vacuole in the yeast cell function in storage, degradation of waste products, and regulation of the cell pH and ionic balance. The plasma membrane in the yeast cell regulates the movement of substances in and out of the yeast cell. Now talking about the yeast cycle, let's talk about the yeast cycle. It happens in two phases, either in haploid and diploid, and both. So in the haploid cells, yeast exists as a haploid cells with a single set of chromosomes, and it is capable of mating. Now the mating types, A and alpha, two haploid cells of opposing mating types A and alpha can fuse to form a diploid cells. In diploid cells, these two haploid cells of two different types A and alpha, as I told in the previous slide, they produce a haploid cells, and these cells contain two sets of chromosome, one from each parent. Diploid cell can undergo meiosis under nutrient depleted conditions. Sporulation and meiosis takes place in the diploid cells. So these diploid cells can sporulate to form 4 haploid spores, each with a single set of chromosomes. Here in the image we can see the haploid cell of A type and a haploid cell of alpha cell can fuse or mate to form a diploid A alpha cells, as you can see here. How the A cells proliferate by just mitosis, making more amount of haploid cells. So now the diploid cell of A alpha, which has formed by the mating of A cell and alpha cell, via mitosis they proliferate on one end, and via meiosis or sporulation these diploid cells form 4 haploid spores. And after germination, these 4 haploid spores can form 2 A cells and 2 alpha cells, which are haploid in nature. And this is how it works. You can see in this image. Now talking about the asexual reproduction that happens in yeast in detail. We talked about the budding process. The primary mode of reproduction in yeast is budding, and we have talked about the process in the previous slide, where a new daughter cells form an outgrowth of the parent cell. How the cell cycle happens, how the cell cycle is controlled, because as you know, in eukaryotic cells the cell cycle is controlled and it is regulated. It has checkpoints, it has regulatory proteins. So how in yeast cell cycle is controlled? The cell cycle in yeast is tightly regulated with specific checkpoints ensuring proper DNA replication and division. It has regulatory genes. Genes like CDC28, which is CDK1 in yeast, plays a crucial role in regulating the cell cycle in yeast. Talking about sexual reproduction, the mating process, as we talked earlier, haploid yeast cells or opposite mating types secrete pheromones that induce cell cycle arrest and preparing the cells for mating. Now, how the haploid cell form diploid cells? The fusion of two haploid cells form a diploid zygote, which can undergo vegetative growth or enter meiosis under specific conditions. There are two methods of reproduction in the image you can see. One is the asexual reproduction of budding and one is the sexual reproduction which is sporulative in nature, which gives rise to 4 haploid cells. Now let's talk about the yeast genome. What is the overview of yeast genome, how it is and how it functions. Now talking about size and structure of the yeast genome, the yeast genome consists of approximately 12 million base pairs and it is organized into 16 chromosomes. Yeast has 16 chromosomes, each containing several hundred to few thousand genes. What is the key feature of yeast genome? It has a compact genome system with relatively little or non-coding DNA. It has very little or less amount of non-coding DNA. High density of genes and a significant portion of essential genes for survival. It has a high density of genes and a significant portion of its genetic material is essential for its survival. How is its mitochondrial genome? In addition to the nuclear DNA, yeast has a mitochondrial genome as well, which is essential for respiration and for the energy generation for the survival of the yeast cell. Now, if we are talking about yeast as a model organism, yeast genome as a model organism for the studying of various metabolic activity and various applications. So, how do we manipulate the genetic system of gene? What are the techniques of genetic manipulation for yeast? First is transformation. It is basically the introduction of foreign DNA into yeast cell, commonly used using the lithium acetate method. The second is homologous recombination. Here, the yeast's natural ability to integrate foreign DNA at a specific genomic loci via homologous recombination. See, talking in detail about the genetic manipulation techniques is beyond this topic and you will be talking about in the forthcoming lectures. Next gene manipulation technique is CRISPR or Cas9, which happens in yeast. Utilization of CRISPR or Cas9 system to introduce targeted mutation or gene deletion in yeast. Next is gene deletion or overexpression. Creation of gene deletion strain or overexpression strain to study gene function is one method of genetic manipulation. Now, while we are talking about manipulation or mutation in the gene system of yeast, let's talk about the types of yeast mutants that arises. What are mutants? When you manipulate the genetic system of any organism, the resulting mutated organism is known as the mutant of the original organism. Okay. So the types of yeast mutant. The first one is the conditional mutant. These conditional mutants are the mutants that exhibit a particular phenotype only under specific environmental condition. How? The first conditional yeast mutant is the temperature sensitive mutant. This mutant can display a conditional phenotype, a type of specific phenotype based on the growth temperature used to study essential gene. You can modify the temperature of the growth of the yeast and it shows at a particular temperature a conditional phenotype. This is known as temperature sensitive mutants. Now is nutritional auxotroph. Yeast cells that require specific supplements for growth using selection markers. Basically, what are auxotroph? Auxotroph are the mutant, are the type of mutant of natural organism which requires a specific supplement for growth. What does it mean? If you provide that particular supplement in the media, then only it will be able to grow. So these kind of conditional mutant are known as nutritional auxotroph and it also exists in yeast. And it is used for the study of supplement for growth and useful in selection markers. Next type of mutant is gene deletion mutants. The first type is knockout. These yeast strains in which a specific gene has been completely removed or knocked out. This allows researchers to study effects of the absence of that particular gene on the cell function and overall physiology. In knocked out type of mutation, a particular gene is knocked out or removed. So we study how the absence of that particular gene, obviously then the gene product, results in how it affects the cell function and overall physiology of that particular cell. The next is gene insertion mutant. This is known as knock-ins. In these mutants, a gene or a reporter gene is inserted into the yeast genome, and this can be used to study gene expression, localization, and function by observing the effects of the inserted gene on a cellular process. Next is the point mutant. The first type is missense mutant. These mutants have a single nucleotide change that result in an amino acid substitution in the protein. This can help researchers understand how a specific amino acid can affect the overall protein function. So here we are talking about the different types of yeast mutants and how they are used in various kind of application, how they are useful. Okay. The next type of point mutant is a nonsense mutant. These have a single nucleotide change that creates a premature stop codon. What is stop codon? Which stops the translation, leading to a truncated protein. This helps in studying the effects of protein truncation on cellular function. Next is a silent mutant. These involve changes in the DNA sequence that do not alter the amino acid sequence of the protein but may affect the gene regulation and splicing. What happens in silent mutant is it is not affecting the amino acid sequence. Means the protein sequence of the protein is not affected, the protein will not be truncated. However, because there is change in the DNA sequence, that will affect in the gene regulation and splicing. So this is how it is used. Next is the overexpression mutant. Under this, the first type is constitutive overexpressors. These mutants have a gene inserted into the genome that is under the control of a strong promoter, leading to higher than normal levels of that particular gene product. This can be used to study the effect of increased gene expression on cellular processes. The next kind of mutant is a suppressor mutant, which is, I think, the opposite of the overexpression mutant. The suppressor of conditional mutant basically. These are the mutants that can rescue or counteract the defect caused by other mutants, often by compensating for the original mutation. They help in understanding genetic interaction and the pathway redundancy that happens in case of conditional mutants. Now let's talk about, we talked about the mutants, how after genetic manipulation, what kind of gene yeast mutants are generated and how they are used in various applications. Now let's talk about yeast genetic screens. Before going into yeast genetic screen, let's understand what exactly is a genetic screen. A genetic screen or a mutagenesis screen is an experimental technique used to identify or select individuals who have the phenotype of interest in a mutagenized population. You have a mutagenized population, like you have, let's say, 10 types of mutants in a population, but you have to select only one mutant. Okay, so it's like a screen, or you can say a filter, which is used to select a particular type of mutant, identify a particular type of mutant. Hence, a genetic screen is a type of phenotypic screen or a filter. Genetic screens can provide important information on gene function as well as molecular events that underlie a biological process or pathway. While genome projects have identified an extensive inventory of genes in many different organisms, in a similar way, genetic screens can provide valuable insight as to how these genes function. Now let's talk about yeast genetic screens. So there are different types of genetic screens. The first one is the forward genetics, using the forward genetics. It involves random mutagenesis, introduction of random mutation across the genome to identify genes responsible for a particular phenotype. It's like a knockout. If you are looking for a particular phenotype and you removed or a particular gene, that particular phenotype is absent, that means that particular gene was useful. What is the screening process in forward genetics? Selecting and analyzing mutant that show an interesting or abnormal phenotype. Next is the reverse genetic based genetic screening. Here involve gene knockout collections, systematically deleting or knocking down specific gene to study their function. They'll systematically knock out single gene and they study the phenotype associated with it. What is the changes it is bringing? If you're knocking out, let's say, X gene, what is the phenotype? What is the result of that particular gene deletion or knocking out? And that is registered in a similar way, and they produce a systematically deleted or knocking out gene specific genes to study their gene function. Gene overexpression, systematic overexpression of genes to observe effects on cellular process or phenotypes. Now let's talk about yeast mating and genetic mapping. Mating and tetrad analysis. First is mating type switching. How yeast mate and how the genetic mapping works. Each cell can switch their mating type through a specialized mechanism enabling them to mate with other haploids. Next is tetrad dissection. Following meiosis, a diploid yeast can produce 4 haploid spores, which can be dissected and analyzed individually. Next, linkage and mapping of gene. Using tetrad analysis to determine the genetic linkage of different genes and map their location on the chromosome. And this is how linkage and mapping genes are produced. Now let's talk about yeast epigenetics. What is epigenetics or epigenetic changes? So epigenetic changes are the genetic modification that impact gene actively without changing the overall genetic or DNA sequence. So how the epigenetic regulation happens in yeast? First is the chromatin structure. Yeast chromatin is composed of DNA wrapped around histones, forming nucleosome that regulate the gene expression. Next is histone modification. Chemical modification like methylation and acetylation of histones impact chromatin structure and gene activity. A next method of regulation is gene silencing. Mechanism like the formation of heterochromatin, example at the telomeres, lead to the silencing of specific genes. Non-coding RNAs. Small RNAs in yeast contribute to the regulation of gene expression through RNA interference or RNAi mechanisms. Now let's talk about in detail about the yeast cell cycle, how it is regulated, what are the checkpoints, etc. Let's talk about the cell cycle regulation in yeast. What are the key regulated in yeast cell? First is CDC28, the cyclin dependent kinase or CDK that drives the yeast cell cycle. Next key regulator is cyclins. These are the proteins that regulate the activity of CDC28 throughout the cell cycle. Now, what are the checkpoints in the yeast cell cycle? Specific points in the cell cycle where progression can be halted if conditions are not favorable. Like for example, they check for DNA damage, right? So these are the checkpoints which check whether there is some damage or there abnormality, and so that the further cell cycle is stopped. Phases of the cell cycle in yeast, like eukaryote, it has G1, S, G2 and M phases. The cell cycle has 4 phases with specific cyclin and CDK active at each stage. Here you can see the cell cycle stage. At the G1 stage, you can see the microtubule, the nucleus and the centromere spindle pole bodies. You can see that. When the budding starts, you can see the mother yeast. When the budding starts, the daughter bud is formed. This is the plane of division. And slowly the microtubules elongate on both the side. At the metaphase, you can see the microtubules in the daughter bud as well. And in the anaphase, the nucleus start dividing. You can see the centrosome at the one in the daughter cells and one in the mother yeast. And in the at the end of the telophase, there you can see the divided nucleus with microtubules and centromeres in 2 yeast cells which are still together, and the G1 phase they are separated as 2 mature yeast cells. Now let's talk about yeast and its signal transduction. What are the different signaling pathways in yeast? First is the MAPK pathway. The mitogen activated protein kinase or MAPK pathway are critical for transmitting signals from the cell surface to the nucleus. It is involved in the processes like mating, osmoregulation, stress responses. Why we are talking about signal transduction in yeast? Because we will be using, first of all, we are studying yeast genome so that it can be used as a model organism. So if you are using any organism as a model organism for study of the various processes, you need to understand the various metabolic pathway, the signal transduction happening in that particular organism. That is why we are studying the signaling pathways in yeast. The first one is MAPK, as we talked about, and it is involved in, again I'll tell you, like meeting osmoregulation and stress responses. The next is the pheromone response. Yeast cell use pheromone signaling to detect and respond to the potential mating partner so that two haploid cell can form one diploid cell. The stress response pathway. The pathway is activated in response to environmental stresses like heat shock, oxidative stress and nutrition deprivation. Now let's talk about the yeast metabolism. The central metabolic pathways in yeast are: the first is glycolysis, next is fermentation, and then respiration. In glycolysis, the breakdown of glucose to pyruvate happens, which results in the generation of ATP and precursor metabolites for further cycles. In fermentation happens in anaerobic condition. The anaerobic process where pyruvate is converted into either in ethanol and CO2, and this pathway of fermentation is used in brewing and the baking industry. Next is the respiration. Respiration in yeast takes place in the aerobic condition or in the presence of oxygen. In aerobic process, the pyruvate enters, the pyruvate which is formed in the glycolysis enters the mitochondria and it is fully oxidized to CO2 and ATP is released. Now let's talk about the applications of yeast cells in various fields. So talking about yeast and diseases, yeast models for human disease. First, let's talk about neurodegenerative diseases, how yeast is used. It is used in Parkinson's diseases. Yeast model have been used to study aggregation of alpha nuclein, a protein implicated in Parkinson's. It is also used for the study of Huntington disease. Yeast has been used to explore the effects of mutant Huntington protein, providing insight into cellular toxicity and aggregation. Yeast is also used as a model organism for cancer research. Yeast is employed to study fundamental processes like cell cycle regulation, DNA repair and apoptosis, which are often dysregulated in cancer. What happens in cancer? It is irregular and uncontrolled growth of cells. That means the cell cycle is not regulated. Okay, so yeast is very good model organism to study these fundamental processes for cancer research. It is also used for screening for potential anti-cancer drugs using yeast models that overexpresses oncogenes or have deletions in tumor suppression genes. So yeast models which have deletions in tumor suppression gene or have overexpressed oncogene can be used as an excellent model organism for the screening of potential anti-cancer drugs for the cancer treatment. Next, yeast is used for drug discovery. How? Yeast simplicity and genetic trackability makes it an ideal model organism for high throughput screening of small molecules and drugs that might be effective against various human diseases. Yeast models are used to identify compounds that can rescue disease phenotypes or target essential pathways conserved between yeast and humans. Now, what are the applications of yeast in biotechnology? As I have mentioned earlier, it is extensively used in brewing and baking industries. Saccharomyces cerevisiae is a primary yeast used for the production of beer, wine and bread. The process of fermentation by yeast converts sugars into ethanol and CO2 with application in various food and beverage industries. Genetic engineering of yeast strain can be used to enhance flavor profiles, fermentation efficiency and stress resistance for industrial processes for the production of beer, wine and bread. In industries, yeast is also used for biofuel production. How? Yeast can be engineered to convert biomass into ethanol, which is used as a renewable biofuel. Development of yeast strain capable of fermenting non-traditional substrates like lignocellulose biomass for ethanol production is extensively researched. For the production of recombinant proteins, yeast is used to produce therapeutic proteins, enzymes and vaccine due to its ability to perform post-translational modification. Examples include the recent COVID vaccine. One of the COVID vaccine has used yeast as a model organism for the production of that particular vaccine. It is also used for the production of insulin. It has used for the production of hepatitis B vaccine and various enzyme for industrial applications. Yeast genetics and synthetic biology, how yeast is used for synthetic biology based research and application. It is used for pathway engineering. What is pathway engineering? Tweaking the original pathway into some other pathway or engineer it to produce a particular compound or product. So introduction of entire biosynthetic pathway into yeast to produce valuable compounds such as pharmaceuticals, biofuels and bioplastics. The examples include the production of Artemis, the production of key anti-malarial drug in engineered yeast strains. Production of bio-based chemicals. Engineered yeast again is used to produce chemical traditionally derived from petroleum such as lactic acid, isobutanol and fatty acids. Optimization of metabolic pathway to improve yield, reduce byproduct and increase the economical viability of yeast-based production system. Again, yeast is used for genome editing and as for synthetic chromosomes. Development of synthetic chromosome like synthetic yeast genome project where entire yeast chromosome has been synthesized and inserted into each cell. CRISPR or Cas9 and other genome editing tools are used to create synthetic pathways and circuits within each cells, enabling precise control over gene expression and metabolic flux so that it can be used for a particular application. Now, because we are talking about yeast genetic system, there are online yeast genetic databases which can be used to study the genetic system of the yeast or take out the data from it. The first one is SGD, Saccharomyces Genome Database. This primary database for yeast genetics, it provides comprehensive information on genes, proteins, genetic interactions in Saccharomyces cerevisiae. Includes tools for gene annotation, sequence analysis and comparison with other species. Another one is YeastMine, the data mining tool integrated with SGD that allows users to perform complex queries on yeast genomic data. Users can retrieve information on gene function, interaction and phenotypes as well as create custom data sets. Other relevant databases are POMBase, BioGrid and YScaffold. They also gives you information about the yeast genome system. Now, even if yeast is very important, what are the current limitations and challenges associated with it? The first is genetic redundancy. Many yeast genes have paralogs, making it challenging to study gene function through single gene knockouts. Synthetic genetic array analysis is one approach to overcome this particular limitation of challenge. Evolutionary conservation is another limitation. While yeast shares many genes and pathways with higher eukaryotes including humans, there are significant differences. Results from yeast studies do not always translate directly into humans. For example, certain signaling pathways and cellular structures present in humans may be absent or simplified in yeast. Another is technical limitation. Despite advances in genetic tools, certain aspects of gene biology such as complex post-translation modification remains difficult to study. High throughput screens generate vast amount of data requiring advanced computational tools and expertise to analyze. Concluding this particular lecture, let's discuss what are the key points that we discussed today. A recap of the major topics, the unique advantage of yeast as a model organism, we talked about that. We talked about key discoveries made using yeast genetics from basic biology to application. What are the applications of yeast in various industries and research, the ongoing challenges and future direction of yeast research. We talked about importance of yeast genetics, emphasizing on how yeast genetics has contributed to our understanding of fundamental biological processes. We highlighted the role of yeast in bridging the gap between basic research and applied science with implication for health, industry and technology. Thank you very much.